Welcome! In this video we'll go over how to lyse bacterial cells using detergent-based lysis with CelLytic B to extract recombinant proteins under non-denaturing conditions. In addition to the CelLytic B cell lysis reagent, protease inhibitors and DNAse will be used to minimize proteolytic degradation and solubilize large strands of DNA. The latter reduces solution viscosity and improves pellet aggregation during centrifugation. This protocol follows the manufacturer's instructions for the CelLytic B cell lysis reagent. If you're using a different reagent, be sure to check the manufacturer's specifications to ensure compatibility with the steps in this video. At the end of this video, you'll find a materials list and written protocol, so there's no need to take notes while watching. Before we begin, a quick note on biosafety. Since we're working with genetically modified organisms, it's crucial to follow biosafety precautions. Always work in a sterile environment and dispose of any waste in designated containers. Never pour liquids that have come into contact with cells down the drain. For specific guidance, consult with your biosafety officer and follow your local biosafety guidelines. In addition to safety, to maintain the stability of your proteins and minimize protein degradation throughout the procedure, make sure to work on ice. The method consists of three major steps: removing cell media, washing with PBS, and cell lysis. Start by preparing the solutions required. For the absolutely comical buffer ACB, which is the CelLytic buffer with protease inhibitors and DNAse, weigh out 1 mg of DNAse powder and add it to a 2 ml Eppendorf tube along with 2000 microliters of Demi-water, mixed by shaking the Eppendorf tube. Then prepare the 25 times protease inhibitor cocktail by adding 2000 microliters of Demi-water to 1 pellet of protease inhibitor in a 2 ml Eppendorf tube. Vortex to mix until the pellet is fully dissolved. To complete the absolutely comical buffer, combine 40 microliters of the just prepared 25 times protease inhibitor cocktail with 10 microliters of the prepared DNAse stock and 950 microliters of CelLytic B in a 1.5 ml Eppendorf tube. Mix by inverting the tube repeatedly. Prepare the phosphate buffered saline solution by adding 5000 microliters of 10 times PBS to a 50 ml Greiner tube and filling it up to the 50 ml line with Demi-water. Mix by inverting the tube repeatedly. To collect the cells, add the determined media volume to a 15 ml Greiner tube. Also prepare a counterweight for the centrifuge by adding a similar amount of water to another 15 ml Greiner tube. Then place the tubes into a centrifuge on opposing sides to ensure the centrifuge is balanced, and centrifuge at 3000 G for 10 minutes. When the timer is up, remove and discard the supernatant, which is the media, into a 50 ml Greiner tube by pipetting with a 1000 microliter micropipette. Be sure not to take up the pellet into the pipette tip. Suspend the pellet, which contains the cells, in 1000 microliter of PBS. To measure, use a 1000 microliter micropipette and mix by carefully pipetting up and down repeatedly. Transfer the solution to a 2 ml Eppendorf tube using a 1000 microliter micropipette. Prepare another counterweight using Demi-water and a 2 ml Eppendorf tube and centrifuge at 3000 G for 10 minutes. Carefully remove the supernatant from the cell pellet by pipetting with a 1000 microliter micropipette into another Eppendorf tube for waste. Be sure not to take up the pellet into the pipette tip. To the Eppendorf tube that contains the pellet, add 500 microliters of ACB and vortex the sample well to completely resuspend the cells. Incubate the suspension at room temperature for 10 to 15 minutes to fully lyse the cells. Do not shake the tube to make sure not to denature the proteins. After the cells have been lysed, centrifuge the lysate at 21,300 RCF for 15 minutes to pellet the insoluble material. While the sample is being centrifuged, you can also give yourself a little spin. Carefully remove the supernatant that contains the soluble proteins into a 1.5 ml Eppendorf tube. Be sure not to take up the pellet into the pipette tip. The tube now contains the cell lysate that you wanted to make.